Hello everyone, my name is Dr. Shaunav Roy, and today in the Bio-Kul class, we'll be doing the chapter called Biomolecules. So we're doing the chapter Biomolecules. And in Biomolecules, today's topic is amino acids. Today we'll be focusing on amino acids. So this lecture will be on amino acids. So amino acids, what are amino acids? So a protein molecule is basically called a polypeptide. The protein is called a polypeptide. Poly means many. What is a peptide? So we learn about this, what is called a peptide. So to understand what is a peptide, we have to understand what is an amino acid. So the word amino acid, you see amino and there is acid. Amino means there is an amine group. Acid means there is a carboxylic acid group. This is called the carboxylic acid group. Okay, it's called the COO. So C is, that means carbon is bonded with oxygen with two bonds, and there is a hydroxyl group, right? This is a double bond. It's a double bond, and this is a hydroxyl group. This is a hydroxyl group. So carboxylic acid is basically a functional group. It's a functional group. So in chemistry or in biochemistry, functional groups are certain kind of groups which attach to a particular molecule and they give the molecule a certain characteristics. Okay. So for example, if a carboxylic group attaches to a particular compound, then that compound will have acidic property, right? Amino NH2, amino. So it basically makes something as a basic, right? Basic property. So what is an amino acid? How does an amino acid structure look like? An amino acid is made up of, there's a central carbon. This carbon is called alpha carbon. Alpha carbon. This alpha carbon is bonded with, so carbon has four, it can bond with four different kind of atoms, okay? Because the valency of carbon is 4. Okay? The valency of the carbon is 4. So carbon is bonded, you can bond with 4 different kind of atoms, okay? So in case of an amino acid, the carbon bonds with, as I said, a carboxylic acid. We call this a carboxylic acid. And it also bonds with a NH2. Okay, so this is the amino group, and it bonds with hydrogen. And a very, very important thing, it also bonds with something called the R group. This R group, it varies from one amino acid to another amino acid. So basically, this R group is the group which gives a particular amino acid its particular property. Now, there are 20 types of amino acids. And these 20 different amino acids, they are as a result of the varying R group. So this varies from one amino acid to another. R group can be as simple as H, if we see in glycine, and it can be very complex also. So this is basically the structure of an amino acid. So amino and acid, that's why it's called amino acid. This carbon is called the alpha carbon, which is bonded to all these different kinds of functional groups. So that means this part, this is the common part. Okay. This is the common portion of the amino acid. This is same in all amino acids. This portion, it varies. So now we will look at the different types of amino acids, the 20 different types of amino acids we will look at. Okay. So the 20 different types of amino acids, as you can see, we can classify the amino acids into 4 different categories. What are the categories? We have non-polar. Non-polar means it has no affinity, no attraction for water molecule. Hydrophobic, so non-polar amino acid. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have all these 10 amino acids which are non-polar. 3 amino acids are, so 10 amino acids non-polar. That means their R group, this is the R group. You see, this is the R group. Again, when we look at the amino acid, we can see this is the common portion of the amino acid, the C, which is the alpha carbon, and then we have the NH part, the amino part, the COO part. Now, you must be wondering why it is NH3 here and why it is COO minus. So this is the ionized form of amino acid. This is the ionized form of an amino acid. What is the meaning of the word ionized? In a cell, inside a cell, amino acids are in the ionized form. That means this is the alpha carbon. It is bonded with a COOH. It is bonded with a NH2. It is bonded with a H and a R, right? So this H is lost, and when the H is lost, this becomes minus, right? It gains a negative charge, right? And this NH2, it gains a hydrogen. So this becomes positively charged, this becomes negatively charged. So after this thing happens, the amino acid can be drawn as COO minus, and this should be NH3 plus. This is H, and this is R. This R keeps varying okay, from one amino acid to another amino acid. It can be as simple as glycine, where R is just H. It can be CH3, a methyl group. In case of alanine, it can be CH3CH3. In case of valine or valine, it can be there can be a sulfur in case of cysteine. Okay, it can be cyclic in case of proline. You see, it's cyclic. It can be leucine. This is leucine, isoleucine. Okay, so these are isomers. That means they're having the same molecular formula but different kind of arrangement. Okay, and then we have the methionine, tryptophan, phenylalanine, which has the aromatic side chain. Then we come to the positively charged amino acids, which are also called the basic amino acids. The basic amino acids. Okay, so they have basic properties. So we have lysine, this is the entire R group. Arginine, again, this is the R group. This part is the common part. And then we have histidine, right? We also have the polar amino acids. That means they are hydrophilic. That means these are having affinity for water. Okay, they have affinity towards water, right? So we have serine. So when you look at the hydrophilic ones, hydrophilic amino acids or polar amino acids, you can see that how they have an hydroxyl group here. They have a hydroxyl group which has the polar property, which gives the polar property, right? And also they have a carbonyl group here, which gives the polar property, right? Polar means they are attracted towards the water, right? And finally, we have the negatively charged amino acids, which have the acidic property, right? Acidic amino acids. So we have aspartic acid. This is the CH2CO. Look, this has the acid, right? CH2. This has the carboxylic acid part. Also, we have the CH2CH2COH. Okay, so we have aspartic acid, we have glutamic acid. Glutamic acid has one carbon extra than the aspartic acid, right? This is the common portion, and here in this portion, it has an extra. So it has 3 carbon. This is actually 2 carbon. So now we can understand that if a protein has more of these negatively charged amino acids, so that protein will have acidic property. In the same logic, if a protein has these positively charged amino acids, it will have a basic property or an overall positive charge. If a protein has a lot of the non-polar amino acids, the protein will be hydrophobic. And the same thing, if a protein has lots of polar amino acids, the protein will have attraction towards water, right? So going back to my notes. So now we know what is a central carbon or the alpha carbon. Now we know what's the R of the amino acids. Now we know what is an amino group, what's the carboxylic group. Now we know there are 20 different kinds of amino acids which make up different combinations of protein. So a protein, so we have thousands and thousands of proteins, right? Because there are 20 amino acids which are combining in a permutation combination, and we are making thousands and thousands of proteins. So with the same 20 amino acids, we're getting different combinations of protein. So here we can see how this is the alpha carbon, this is the carboxyl group, this is the amino group. Examples of a hydrophobic amino acid is leucine, as I already showed in the chart. And an example of a hydrophilic amino acid are serine, which has a hydroxyl group, and an aspartic acid, which is acidic, which has a negative charge. Now, we need to understand how amino acids they join together and how it forms a peptide bond, and finally how it forms a protein. So let's explore that concept. So here we have one amino acid. So let's say this is the amino acid 1. And amino acid 1 looks like this. So it has a carboxylic functional group. It has an amino group, H, and let's see, this is R1. This is the side chain R1. So this is now, this is joining with another amino acid, which has, this is the carbon, H plus, COOH, NH2, and this is the R2. Okay. Now this is amino acid number 2. Right. This is amino acid number 1. So this reaction is called the dehydration reaction. We call this as the dehydration reaction. What is dehydration reaction? So now we have the OH from this. So in fact, we can draw this in a easier form. Let's draw this as NH2. Now, a hydrogen from this amino group and a hydroxyl from this amino acid 1. So a hydrogen from the amino acid 2 and a hydroxyl from the amino acid 1 is taken away. And this comes out as water. This is the dehydration reaction. This is the dehydration reaction taking place. What is happening here? So this leads to formation of NH2. This is the first amino acid. This is the side chain CO, or we should say this is actually CO, right? And here we have the NH. Remember, the OH and the H is gone. So this is H, this is R2, and COOH. So this orange box here, this orange box is called, so this bond which is formed is called the peptide bond. This is the peptide bond. So what is the peptide bond? CONH. CONH is called the peptide bond, and a water molecule is released by the process of dehydration. Now, so here we see 2 amino acids binding. There can also be a third amino acid. So basically, there can be a third amino acid. So let's say we have a third amino acid, which is again having the N, H. This is the alpha carbon. This is another side chain R group. And here we have COH. So again, we'll see the same concept. Again, we'll see a dehydration reaction taking place. Okay, there is release of the water molecule. And here, this will change now. So here we can draw this in this form. CO and now the brown amino acid here, NH. This is the CH. This is the R3. This is the CO. OH. So again, we have the formation of, here is the orange box, which is the peptide bond. We'll say PB, peptide bond. All right. So now this becomes a dipeptide. Dipeptide. In this way, another amino acid can join and form a tripeptide, fourth amino acid to form a tetrapeptide, pentapeptide, okay, like that, okay. A protein is formed like this. A protein has thousands of amino acids. So a protein which has, so if you draw a protein, we can draw a third bracket and we can put N, where N can be 1,000, right? Or even more than that. It can be even 10,000. Okay. So that's why a protein is called a polypeptide. A protein is called a polypeptide, which has these thousands of amino acids which are joined together by dehydration reaction, right? So there are many peptide bonds. Okay. Now, the notes will tell you the same thing. If you follow my notes, it has the same information. The question is that we know that how a protein is formed. Now we learn how a protein is broken down. Okay, so this is called the hydrolysis. So with the help of a water molecule, a dipeptide, a tripeptide, it can be broken down. So let's see how the protein destruction or protein breakdown taking place. So this is called the hydrolysis reaction. Okay, so we are going to study the hydrolysis reaction. Okay. So what is happening in hydrolysis? Hydrolysis is a reaction in which a molecule is broken down with the help of a water molecule, right? So again, let's draw our amino acids. So this time we have a dipeptide, okay? So we have a C, H, CO, NH2, and here is our peptide bond, N, H, alpha carbon. This is the H, this is the R2. So I forgot to draw the R1, right? This is the R1. And then we have C, O. Let's go for the third one. Maybe it's a tripeptide. So now we have N, H, C, H, R3, CO, OH. Let me draw the orange boxes for the peptide bonds. Now, as I said, this is a hydrolysis reaction. This is a hydrolysis reaction. That means water is used to break down this thing. So now, whenever we're adding water, it's the reverse thing happening. OH is going to the carboxylic group and 1H is going to the amino group. OH is, again here, OH going to the carboxyl group and 1H is going to the amino group. So we are back to what we started with. NH2, CH, R1, COOH. This is the first amino acid. And then we have NH2, CH, R2, COOH. And we have the third amino acid coming up, where we have NH2, C, R3, All right. So this is a hydrolysis reaction in which a protein is broken down. So we learned today what is amino acid. So let's just try to summarize today's class. So we learned so this is a hydrolysis reaction. As you can see, the peptide bonds, how the peptide bonds are broken, and they're back to their amino acids. Okay. So today what we learned about what are the amino acids. We learned amino acids have an amino group and a carboxyl group, and there's an alpha carbon, there's a side chain. Depending upon the side chain, we have 20 different kinds of amino acids. We have amino acids which are non-polar, having a non-polar hydrophobic side chain. We have amino acids which are positively charged, which give the basic property or a positive charge to a protein. So the R group or the side chain has a positive charge group, and then we have a polar amino acid, that means hydroxyl, or we have a carbonyl group, and that makes the amino acid having attraction towards water, hydrophilic property, water loving property, and finally we have a negative charge R group, which is basically acid. So we have aspartic acid and glutamic acid. So we have 20 different kinds of amino acids based on the side chain, right? Side chain can be as simple as a hydrogen, like in glycine. Okay, it can be as complex as in arginine, with several kind of carbon, right, joined together with amino group, right? So this is very much complicated compared to the glycine. We also learned about the dehydration reaction and how the two amino acids, they join together, lose water, and they form the peptide bond. And we also learned about the hydrolysis reaction, how the peptides are broken down into again subsequent amino acids. So this is all for today. Again, watch my Bio-Kul classes for more content, and in the next class we'll come up with more interesting content. Wish you all the best, and bye-bye.